And then once you hear that, you're good to get started.
Okay, thank you, Julia. Good afternoon, everyone, and welcome. This is the 67th annual Gilbert Newton Lewis Memorial Lecture, but the very first Lewis Lecture presented virtually. So we are all a part of some history here today. And it is my great pleasure to introduce our renowned colleague, Birgitta Whaley, as this year's Lewis Lecturer. This distinguished lectureship was established in 1954 and is held annually in October to commemorate G.N. Lewis's birthday. Remarkably, Lewis served as Dean of the College of Chemistry from 1912 through 1941. That's one record that will surely never be broken. And he was largely responsible for the college's rise to fame. Lewis's career spanned half a century, a period widely regarded as one of the most brilliant times in the history of scientific discovery. And his name ranks among the highest on the list of those who made it so remarkable. The electron theory of chemical valence, the advancement of chemical thermodynamics, the separation of isotopes, and the generalized theory of acids and bases are among the achievements that will forever be associated with his name. Associated with Birgitta's name are many achievements in theoretical chemistry and quantum physics. A professor of chemistry, Birgitta is also a faculty scientist at LBNL and co-director of the Berkeley Quantum Information and Computation Center. She received her bachelor's degree from Oxford University in 1978, spent 1 year at Harvard on a John F. Kennedy Fellowship, and earned her doctorate in chemical physics from the University of Chicago in 1984. She was a postdoctoral fellow at Tel Aviv University and the Hebrew University of Jerusalem prior to joining the faculty here in the Department of Chemistry in 1986. As one of the world's most interdisciplinary theoretical chemists, Birgitta's research program lies at the interface of chemistry and quantum physics and at the joint interface of these two disciplines with biology. Her work focuses broadly on quantum information and quantum computation, on control and simulation of complex quantum systems, and on quantum effects in biological systems. Her many important contributions include developing algorithms allowing quantum computers to efficiently solve computationally complex problems, including those of quantum chemistry. And she has played a key role in the development and application of quantum control for the realization of physical schemes for quantum information processing. Birgitta has authored over 240 scientific publications throughout her career, and her work has been widely recognized with numerous honors and awards, including, to name just a few, the Alfred P. Sloan Award, the Alexander von Humboldt Senior Research Scientist Award, and several visiting professorships in the US and abroad. She is a fellow of the American Academy of Arts and Sciences, the American Physical Society, and the International Academy of Quantum Molecular Science. And as another testament of her scholarly prestige, Birgitta currently serves as one of 12 members on the President's Council of Advisors on Science and Technology. Birgitta has also shown outstanding professional and academic leadership over her career, and she has proven herself to be one of the campus's most dedicated citizens. She has served on important academic senate committees and on several chancellor's advisory committees. And since 2014, Birgitta has served on the board of directors for the Women's Faculty Club. Her commitment to her colleagues and to furthering the mission of Berkeley's campus is highly commendable and downright inspiring. So now it's time to turn things over to Birgitta, who is going to tell us about her recent research in quantum biology, which seeks to characterize and understand the role of quantum dynamical effects in biological systems. So without further ado, please join me in welcoming our distinguished colleague and the 2020 Lewis Lecturer, Professor Birgitta Whaley.
Thank you, Doug. Thank you, Doug. Okay, is that good? Is that visible now? Okay, so it's a great pleasure to be here today and to give this lecture in honor of G.N. Lewis. And so this little path going through these beautifully green woods will sort of be, in a sense, a metaphor, what I'm going to do today to show you how we think about quantum effects in biology, which may seem somewhat paradoxical paradoxical if you're not used to thinking about quantum mechanics, particularly in biological systems. So let me first, let me first pay homage to Gilbert Lewis. So G.N. Lewis actually has an intimate connection with my talk because he is responsible for the term photon for light, and he proposed it as a term for radiant energy in 1926 in a letter to Nature, and this was picked up really surprisingly, not immediately, but a couple of years later by de Broglie and Compton. And then by 1928, it entered the Index of Physical Review. And by 1930, Dirac had put it in his book. And this name is now a household name for our notion of light coming in small packets of energy. I think, however, though, G.N. Lewis would have been, when he envisaged this, this term of photon for describing little packets of radiant energy, he actually didn't like Einstein's idea of light quanta. He didn't apply it to light itself. He applied it to energy inside an atom. And he would have been quite surprised by the picture on the bottom right, which I think encapsulates it from a recent paper in Nature Photonics, showing how single photons can be emitted from carbon nanotubes when they are, in a sense, activated by having a defect. This is an aryl species providing a defect in the wall of the carbon nanotube here. And when this is irradiated with sort of visible light in the red, it causes causes photo emission from the nanotube in the telecom wavelength region. And this picture nicely encapsulates our modern view of photons, namely that they're a small packet of an oscillatory motion of the electric field associated with light traveling through space. And on the left-hand side is written a wave packet. The form Xc of t is the wave packet. And so what I'm going to be talking about today is wave packets described here in mathematical language, which contain, if you look here on the left, contain exactly one photon of light. And before I go on, can everyone see my cursor, or should I use it annotate? It's good.
I think we can see your cursor, so it's good.
Okay.
We can see it.
So I'm waving it around. Okay, good. So then I'll continue to use that rather than using annotate, because if I use annotate, then it's hard to advance the slides. Okay, so what is quantum biology? Well, actually, this is something which, pardon the pun, has long roots. It was first really discussed by Bohr in about late 1920s, and his student, Pascual Jordan, who started turning their attention to biology and to how quantum mechanics might be playing a role or manifesting itself in living objects rather than the inanimate objects of atoms and small molecules that they'd been looking at before. And so they wrote some articles which were primarily concerned with complementarity, of trying to find some analog of a complementarity principle between waves and particles in physics, trying to find some analog of that in biological systems. And nothing really much came of that, except maybe the first paper with the term quantum biology in it, which Jordan published in 1932. Bohr carried on. In fact, in 1960, he published another paper about life and light. But the person who really took the link between quantum mechanics and biological systems seriously and made the first really serious study is Max Delbruck, shown here on the right, who was a quantum physicist. And he did the right thing, namely, rather than theorize with some like-minded colleagues from the same discipline, he went out and crossed the street and went to talk to a geneticist, Timofey Vrosovsky, and Kurt Zimmer, who was what was called at that time a photobiologist, and nowadays would probably be called a chemical physicist. And they undertook a series of combined experimental and theoretical studies using X-rays to probe what at that time was known as the genetic material, and studied the mutations of these. And from those studies, they came to some conclusion about the stability of the genetic material. And this was long before the structure of DNA was established, so they didn't really know what they were dealing with in molecular terms. So this was really, I would say, the first quantum probe of biological structures and their function. And it was an acknowledgement, it provided an acknowledgement of the need to understand the detailed molecular structure of functional biological entities. So since then, I tend to divide the sort of growth of quantum biology as a topic into two eras. So these eras are named BL and AL, which may be obvious to you. I'll say in a moment what the L is. So the first era really encompasses the study of molecular structure, pathways to chemical reactions, the transformations of molecules, so energetics, questions of energetics, kinetics, and stability. And what was involved in here was really understanding how the quantum nature of molecular energy levels came into play in influencing the stability of biological systems and energy barriers to transformations between them. And this was really summarized in Schrödinger's, first of all, an article in 1943, and then his book in 1946, which was strongly based on the work of Delbruck and his co-workers on the X-rays, studies of the genetic material. So the second era was from the 1960s onwards, and this tells you probably that L is laser. So with the advent of lasers, people could now study quantum dynamical effects, and that led to new generations of dynamical probes, and then increasingly over the last 20 years or so, new innovations technically via quantum science and technology. And this is a picture from one of Graham's articles, which I'll talk about a little later on, where they're probing with a laser here, coming in with these little pulses here at regular intervals. They're probing a light harvesting system and learning information about the excited state dynamics. And this is emulating, this laser here is emulating the activation of this process by sunlight. So in that first era, in red here, all these things here, so I've listed here sort of a historical survey of major sort of steps forward in understanding of quantum behaviors and applications to atoms and molecules. And in Schrödinger, by the end of this first era, or certainly by 43, when Schrödinger was writing, there was the entire structure and statistics was here. The quantum statistical theory of chemical reaction rates was very important, and contributions from Eyring and Polanyi and others. But there was no mention of tunneling. So even though tunneling was very well known, Gamow first wrote about tunneling in 1928, and there had been this very beautiful work by Bell in 1933, looking at hydrogen tunneling in chemical reactions. There was no mention of tunneling at the time. And there was also no mention of coherence, quantum coherence. In particular, no mention of the kind of non-local quantum correlations, which are Schrödinger referred to as entanglement, which is regarded as one of the defining characteristics of a quantum system rather than a classical system. So, and part of the reason for this was that there really was no evidence for this at the time, no experimental evidence. So what I've put on this slide is now a sort of overview of the kind of topics where people are looking for quantum effects now. And most of these topics concern specifically these kind of features that were not accessible before in the earlier days of the first era. So what I'll focus on today is the photosynthetic system in plants and bacteria, where quantum coherence is involved. And there's a lot of experimental evidence of this here, which is one reason why I'm going to focus on it. But there are other reasons, other areas where coherence has been invoked, ion channels, certainly in vision. And this is actually work from Rich Mattis, one of his papers, where coherence and non-Born Oppenheimer dynamics are very important in the initial stages. So both, then there's
Thanks.
entanglement as more exotic kind of non-trivial quantum effect. And this has been invoked in the bird navigation phenomenon, which is also like vision and photosynthesis. The mechanism by which birds are believed to navigate around the globe and travel long distances is something which is initiated by light, one of several mechanisms that they have. And then there are other examples where people are looking at tunneling in animal smell, for instance. And then there's finally the brain, which I'll say very little about because there's really very, very little experimental evidence. So I can make some comments at the end if people are interested on that. So these are a wide range of topics. And as I said, 3 of these topics involve activation by light. And I'm going to focus on the photosynthesis now. And so I've said that in the second era of quantum biology, we had these tools since the laser, in particular the ultra-fast spectroscopy, which has proven to be very important for understanding electronic energy transfer in photosynthetic systems. And there are other new tools coming on board here. But now I think we're moving from the second era into maybe a Mark II of the second era or even a third era, where we can look at quantum light sources, meaning sources that produce reliably on demand single photons, even entangled photon pairs, and one can do photon counting. So that's the new era, which I'll tell you about in the later part of the talk. So let me now turn to photosynthesis. So here's a very brief summary for those that are not familiar with it, of what's involved in photosynthesis. And what's involved is shown in this cartoon on the left, namely that light is absorbed by a photosynthetic system, which could be green plants, it could be bacteria near the surface of the earth, here in a shallow pool, or it could be bacteria. These are bacteria living near what are called black smokers, about 2 kilometers below the surface of the oceans. And these bacteria use infrared light, because of course there's no visible light that penetrates to that depth. So the principle of photosynthesis is that light is absorbed by some molecular system inside the plants or bacteria. And the light, the first step of the photosynthesis is the absorption of light. And then there's a process by which there's a decrease in energy so the light travels across an antenna in space, and it also generally lowers its energy until it gets to a particular part of the structure, which is known as a reaction center, where the excitation separates into an electron, which is free and moves off, and a hole, which is then neutralized. And this is followed then, this charge separation is followed by real chemistry, which is the so-called dark reactions, the secondary electron transfer reactions involving water splitting, proton transport, and etc. And then formation of ATP and basically storage of energy, then of the energy in chemical entities. So what we're interested in is this very first step, namely light is absorbed and then transferred across to the antenna system to the reaction center. And this first step is known as the light reactions, is characterized being not only rapid, but it's also very efficient. And it leads to approximately greater than 95% conversion of photons under weak light conditions. So this is summarized here for the photosystem 2, often abbreviated as PS2 system, which is responsible for photosynthesis in about 50% of green matter on Earth. This is very important super photosynthetic complex. It's known as a super complex because it consists of many small subcomplexes shown here. And the outer ones here in yellow are the antenna, so-called antenna systems, where the light is primarily absorbed. And so, for instance, the light could be absorbed at this location here, and then it has to travel in space across about 10 or 20 nanometers to get to the reaction center in here, where the charge separation occurs. So the time scale for this transfer of excitation is about 200 picoseconds to go from here to just the initial stages of the charge separation. And this transfer of excitation occurs with very high efficiency. So in the second era, which takes us up until about a couple of years ago, really, ultra-fast spectroscopic techniques, in particular, the multidimensional spectroscopy techniques developed by Graham Fleming and his group and co-workers. So firstly, the two-dimensional electronic spectroscopy, and then secondly, more recently, the electron vibration spectroscopy have really given us a lot of insights into the nature of the excitonic energy transport once a photon is absorbed. And the idea of this spectroscopy is that you put in basically here 3 pulses, and the first pulse creates a coherence. The second pulse puts the population in the photosynthetic light harvesting system up into the excited state. And then you wait for some time and you put a third pulse in, which brings you back down to the ground state. And then you plot, then you Fourier transform with respect to these time delays here, this one here, and the last one over here, between the third pulse and your detection. And that gives you a two-dimensional plot in frequency space. And if you do this as a function of this waiting time in the middle, the capital T, this spectrum will change, and the way it changes will tell you about the dynamics when the system is in the excited state. And here's an example from Graham's very famous paper from 2007, which was this example that I showed was previously on that beautiful schematic, which takes part of the so-called FMO protein, which is a small protein which connects a big antenna system to the reaction center in a bacterial photosynthetic bacteria here, which is a very unprepossessing bacteria, lives in these shallow pools. And so here's a big antenna where the light is actually absorbed. And then this protein sits between the antenna and the reaction center and the excitation. So by the time the light energy gets to the FMO protein, it's already in the form of an exciton, and it then moves through this protein. And so what Graham and his group did, they made these two-dimensional plots, two-dimensional studies of the electronic spectroscopy. And so if they did this, this was at a particular time, waiting time capital T, 200 femtoseconds. And then if they repeated this for many different waiting times, they would find this three-dimensional figure. And what you see here is these oscillations as a function of this middle time, T2, they tell you about the coherence of the dynamics in the excited state. And so there's been many studies since then that show that that replicate this and also show it exists in other photosynthetic systems. And basically they reveal a lot about the interplay between the excitonic degrees of freedom and the vibrational degrees of freedom in making this energy transport through the FMO protein happen. Okay, so that's still a very active area of research, and there's a lot of very important questions about to what extent the vibrations contribute. But now, and also people had there's a lot of debate about whether this coherence, which has been clearly observed in these studies, whether it's important for the quantum efficiency. So I've mentioned that this quantum efficiency is very high, it's 95 to 98%. And it was rather surprising to me when I first started working in this area that you can actually go online and you can find a picture and you can, for a quantum meter that you can buy from Amazon to measure the photon flux in your garden. And so that basically told me that there's something maybe a little bit fishy in this definition of quantum efficiency, which is that namely that these measurements are highly averaged and they're made, essentially they're basically kinetic measurements made by on macroscopic samples. But the interpretation is always given as one photon, so one light quantum, is transduced to one electron hole pair. So the interpretation is always a microscopic interpretation. But no one really knows whether that's true, that microscopic interpretation. So we're in the situation that the chemical dynamics community was in the maybe late 1970s, before molecular beams, where we knew a lot about kinetics and mechanisms of chemical reactions from bulk studies, basically kinetic studies. But with advent of molecular beams, we could actually then go and look at individual collisions between molecules and see how the individual collision would redistribute the energy between the internal degrees of a molecule. So we want to do something very similar to, or analogous to that, for the photosynthetic quantum efficiency. In particular, we want to understand what is the mechanism and the timescale for the absorption of one photon energy equivalent from sunlight. We need this in order to be able to make a microscopic to actually validate this microscopic interpretation. So let's think about how is sunlight absorbed. And on the left, I have a picture with a little summary underneath saying, so what do we know about sunlight? Well, we know it's essentially black body radiation, but strongly filtered by the atmosphere. We know it's also ultra weak intensity. So if you go and actually calculate how much, what's the number of photons per mode or per frequency mode, it's very, very low. And there's also, somewhat surprisingly, very weak coupling to chromophores. So in a sense, this weak coupling to chromophores provides a sort of bottleneck for getting light into the photosynthetic systems. And there's all sorts of reasons we could talk about later about why that might make sense in an evolutionary point of view. But let me continue with rather than the why, I'm going to continue with the how. So we can think of a light harvesting complex as a photon detector with a very high internal quantum efficiency rather than the external. So this is in an engineering sense. Internal quantum efficiency says, what is the efficiency after the light has been absorbed? What is then the efficiency of taking the absorbed light, which is now an excitation, and generating electron-hole pair? So we know that the detection involves these three processes: absorption, energy transfer, and electron-hole pair separation. And we have a feeling that from all the experiments done with ultrafast spectroscopy, that coherence is important, at least in the EET part of these processes. But what we don't really know is much about the photon absorption event, the first part here. So for this, we have to then think about what is sunlight. And we're going to take a single photon description for individual modes and try to understand in what manner a light harvesting complex builds up enough energy to release a single electron. And so we suspect this might require many photon absorptions. And there's certainly time for this because the turnover time for the PS2 is about 4 milliseconds, which is a very long time scale on the time scale of exciton energy transfer. But we suspect we might need a lot of photon absorption events because the probability of absorption of a single photon by any individual chlorophyll is very small. It's about 10 to the minus 6. And let me illustrate that here. Also with the title here that the chlorophyll absorption is making a measurement of a single photon, but it's a very weak measurement. So on the left, I've shown the absorption spectrum of a bacteriochlorophyll A. That's this, and what's shown up here is just the head of the molecule, which is the part that does the absorption of light. And there's several bands here, and we're going to focus on the QY band. And there's a bandwidth associated with this. And so if one thinks about a chlorophyll as a two-level system, so then one sees that this probability of absorption, 10 to the minus 6 is down here. And this is the absorption that one expects. So these are theoretical calculations done with coherent light, like from a laser. And these two lines here, one of them indicates calculation done with this particular wavelength taken from the experimental absorption spectrum. And the other one is the line, if you take the bandwidth of the visible light, which would be from about 400 to 700 nanometers. So that's a big difference on this picture here. But then if you look at this logarithmic scale here of bandwidth, it's very, very close. And this is plotted on a logarithmic scale to show you how far away this absorption is. So even if you were to take an isolated chromophore without worrying about coupling the excitonic, just those excitonic levels, without worrying about coupling to the phonon levels, you see it's very far away from the ideal absorption of that two-level system, which is of order 1. It's about 48%. And that's at a very different, much more narrow bandwidth. So essentially, this tells us that even without the vibrational degrees of freedom of this molecular chromophores, that the resonance is relative to the ideal two-level system. It's very far off resonance. And then also, in addition, we have the interaction with phonons. And when we add that in, we see that for all bandwidths, there's a suppression by 5 orders of magnitude or so. And that's not surprising. That's coming from the dephasing from the vibrations. And these values of 10 to the minus 6 are consistent with single molecule absorption measurements, which were made for the first time really in 2011. There were 3 groups using 3 different techniques, measured the absolute absorption probabilities of single molecules. And they are also, for similar molecules to chlorophyll, they're of the order 10 to the minus 6. So that's all consistent with this. And down here on the left, this is actually a very interesting estimation that my student Li Wen Ko has made recently, saying that if you are having a very narrow, so in other words, if you have large bandwidths over here, you have a very narrow pulse. And so if you take a single photon pulse with this bandwidth, you're in a regime where the probability of absorption is given essentially by the ratio of the spontaneous emission rate to the pulse bandwidth times essentially a universal function f of the ratio of the whatever energy variation you have, whether it's an energy variation in a complex, like different energies. So this is inhomogeneous broadening in the chemical language. So static energy variation being inhomogeneous broadening, and the dynamic would be the homogeneous broadening due to vibrations. So it turns out when you put the numbers in for chlorophyll here, you find out that for a typical light harvesting system like the antenna complex of PS2, which is called the light harvesting complex LHC, you get about 10 to the minus 6. So this is all consistent. So, okay, so we're going to look at single photons and from sunlight, and we're going to use this idea, the notion that single photons from sunlight are coherent. And there's a lot of debate about sunlight, what it is, and whether it's coherent or incoherent. And here's 2 arguments why one might want to use coherent photons to simulate the action of individual photons on photosynthetic systems. The first is if you estimate what's called the coherence volume, which is basically a product of the coherence area of sunlight at the surface of the Earth, which is here, and that's been known for many, many years. It's quite large. And the product of this coherence area with the coherence length, which is essentially a measure of the temporal coherence, and that's basically determined by the bandwidth. So if you consider your molecule, chlorophyll molecule, to be measuring a photon from the sun, so basically it's collapsing the photon from whatever state it was in to something which has this bandwidth, then the respective coherence volume is about 53,000 micrometers cubed. And the volume of a chloroplast, which contains many, many photosynthetic systems, is only 20 micrometers cubed. So this tells us that we are well entitled to consider single photons from sunlight incident upon a photosynthetic system as being coherent. Second evidence, or let's say argument for considering single photons to be coherent from the sun is an experiment done last year by Jian Wei Pan's group in China, a very beautiful experiment, which it was basically a quantum optics experiment showing quantum interference between a photon from sunlight here and a single photon from a semiconductor quantum dot, so completely artificially generated. And the point of this was to show that you can, from my perspective, the point that I take away from this is not that the photons from sunlight are necessarily coherent, but that if you have the right detector, and so you see here, this is what was done to the sunlight to actually generate the photons that were then input into this interference experiment, So if you have the right detector, your photon can be coherent. In other words, your sunlight photon can be collapsed onto a coherent, a single coherent quantum photon. And this interference experiment is called Hongu-Mandel, after the people who first did it. And basically it shows that if you have coincidence, that when you put photons in from these two directions here, you will always see, because of the bosonic nature of the photons, you will always see two photons on one or other of the detectors. And that's indicated by this minimum here in this coincident count of one photon here and one photon there. In other words, you don't see one photon in each of these arms. You see only two photons in either A or in B. So both of these are encouraging for looking at single photons coming from sunlight in the first instance as coherent single photons. And so the new quantum tools which we're using now are basically sources of single photons. In particular, we're using sources of pairs of single photons. One is generally called the idler and one's called the signal. And I'm going to show you how we use these tools to learn something about what happens when a single photon is here incident on a light harvesting system. In particular, what we want to do is study the subsequent energy transfer and as we detect it by single photon fluorescence. And so this quantity down here is going to show up a couple of times. We want to know in particular how many photons are absorbed per single photon fluorescence that's emitted. So here's an analysis, or here's a proposal of the experiment. So this is an experiment that Graham and I talked about for several years, and was then funded a year ago. And I'm going to also show you some really preliminary results about this. The idea is to put our light harvesting system, ideally in the end it'll be PS2, but and I'll show you theory for PS2. We put it in here, and then we basically have two coherent photons coming in here. And one of them is the herald. And when the herald is detected, it sets a timer down here. And then it also opens up a filter here, which allows the signal photon to go in through here. Sorry, that's the idler photon to go through onto the sample. And then we have another detector on the other side of the sample. And if this detector doesn't detect anything, we know that there has been no transmission. And then we look for coincidence with the fluorescence photon here. So the idea is to select firstly a fluorescence proxy for electron hole pair creation. So we might want to look at fluorescence from some chlorophyll down here in the reaction center, given that you have absorption up here. And we'll do this with single photons in a pure state. So the first big question we want to answer is whether we, for one fluorescence event, do we need to have many, many photons, like this kind of distribution here, photons absorbed, or is it possible that the entire energy for the release of electron hole pair gets absorbed in one single jump? Namely, that we'd have a large, significant amount of fluorescence events which happen after absorption of only one photon. And quantum jumps are somewhat disruptive. They were first proposed by Bohr in 1913, well before quantum theory was really fully developed. And they really weren't, many people didn't believe in them. Some people still don't believe in them, but since mid-1980s, they have been seen. Basically, you should think of a quantum jump as a discontinuous change in the state of a particular quantum system due to its being monitored by some means. In our case, the monitoring will be by the detection of photons from fluorescence. So here's a schematic of a theory study of this, where we take two photons. One is the herald that comes out here, and the other one is the signal which goes into the chlorophore system. And so the theory, then we can just regard this as a single photon being incident on our photosynthetic system. And then we're going to look at the relation between photons that are transmitted and photons that are fluorescing. And so here's PS2. So PS2 had about 325 or 24 chlorophylls. They're distributed. This is sort of symmetric structure, a dimeric type of structure. And we divide them up into those in the antenna, which are depicted by green, those are in the reaction center, which are in orange, and then those who are in between, which is in what's called the core complexes. And you see we have these sort of double layer type of structure. If you look at the dipole moments of these chlorophylls, which determine the absorption probabilities, you see that the net, appear quite disordered. The net dipole is relatively small. Each individual chlorophyll has a dipole moment of about 4 dBi. So the net dipole from 324 is only 1 order of magnitude greater than one of them. And when we think about how it interacts with a single photon, we have to take collective emission into account, collective emission and collective absorption into account, for the reason shown here, which is that the sorry, I forgot to disconnect the phone. So for interaction with single photons, the single photon will cover the entire size of PS2. It's known as the dipole approximation in optics. And so here's PS2 relative to the wavelengths of the light. And so then that means that we really have to take into account the collective nature of the dipole properties of this system. And there are well-established ways to do this. And it means that basically we have an emission rate, which is a tensor, which is determined by the product of transition dipoles on different pairs of different pairs of chlorophylls. And this means that when we think about what kind of emission or absorption, this is shown here for emission, properties these systems will have, we have to actually then, to obtain our collective emission operators, we have to diagonalize this matrix of the tensor of the dipole moments. And that will give us a set. So this is a singular value decomposition. This gives us 3 singular values, meaning 3 non-zero values, which are the collective decay rates. And you can think of these as decay rates in three directions, X, Y, and Z, so the 3 directions in space. And it also defines then our collective emission operators. And these decay rates can be in any 3 directions, which we can determine in any arbitrary coordinate system we like. But if we so here in one coordinate system that's related to the structure that I showed you before, you see basically the different chlorophylls that contribute to the X, to the Y, and the Z. And what's interesting is that on the left, you see the decay rates are basically enhanced relative to the decay of a single chromophore. So there's some degree of cooperation in the decay, in the spontaneous emission. And that means also correspondingly, there'll be some degree of cooperation in the absorption because the same operators are involved. So now, so I better maybe skip over this because this is, so basically what we've done is made simulations where we take into account the solid angle of a detector. So these are quite realistic simulations. And we basically are designing our theory to be able to calculate the photon flux going out in a particular detector of a certain solid angle here. And we have then these different measurement operators, which are these are the ones that we might be detecting. So the transmission is along here, and then there along the fluorescence, we would have X and Z polarization if there's input Y polarization. And then everything else is unmeasured. So these are the basic losses of the system. This is one of the only 2 theory slides, and this is just for the series to show you how we actually solve for this. We use what's called a stochastic Schrodinger equation that basically allows evolution of the excited state of the system. And this is a single excitation either in the system or in the field. And we allow an evolution of this under a master equation that is interrupted by these jump operators, which are the operators allowing emission of photons into either the transmitted mode or the side mode, so the fluorescent mode here. And the outgoing modes will be then fed by these operators from decay of an excited state of the chromophore system, beta, which is this excited state here, which is generated by an input single photon. So let me show you without any do some results. And so these are calculations which show the fluorescent count rates, which are the emission quantum jump rates. This is the transmitted on the top here, and on the bottom is the fluorescence. And if you analyze these fluorescence rates, you see that they show oscillations. And so here are the two different polarizations of fluorescence. And on the right-hand side, we have made a Fourier transform and plotted with the little gray lines indicate the energy differences in the chromophores in the PS2 system. And you see that basically these peaks all show up at the lines corresponding to energy differences in the chromophore system. So these peak frequencies correspond to excitonic energy differences. So what we're seeing is that the jump rate, namely the rate of counting of individual photons being fluoresced from the system, are providing a witness of the excitonic coherence. So this is a new kind of witness. It's not really coming from spectroscopy. It's coming from photon counting, which is a different kind of spectroscopy. And if you integrate this, you then find, as you expect, that with time that you get a rise in the number of counts from the, and also we can analyze. So this is all consistent with what we would obtain if we did normal types of calculations where we just integrate the system without measuring any photons individually. So we can also make these individual trajectories. So this was with one pulse. This is one trajectory. And we see that conditional upon knowing that no photons were lost before the jump time, here you see the conditional probability of being in the antenna system. Here's the conditional probability of being in the CP, the blue, and here's the conditional probability of being in the reaction center. And so this is coming from a photon pulse that was absorbed this very short time here, which was centered on the antenna system, but it has sufficient bandwidth that it also can excite a little bit of the reaction center and a little bit of CP. And then this trajectory at some point here, about 9 picoseconds, everything drops in the excited states and the system goes back to the ground state. And that indicates the location of a quantum jump. And if we run more trajectories, we'll see the jumps occur at different times. So that's all well and good, but that was an idealized set of calculations that doesn't have, that only has the chromophores and has no phonons. And we know from the ultra-fast spectroscopy that phonons are very, very important. So what do? So what we've done is to add phonons, and we do it in a standard, this picture is one that's very common for this area, literature. So we're basically adding coupling of the excited states to a phonon vibration here, UM, and there's a whole set of these for each site, XC. And then we do electronic evolution to the second order. This is a non-local differential equation here. And if you think about, so schematically, if you think about Fermi's golden rule, then you think that you will realize that there's going to be some perturbations here, V, and these perturbations are being induced by this coupling. And so this, if we say this V is something that projects onto each of the sites, then we can get three kinds of jumps from this. One is a heating jump where the energy goes from I to J, and J is greater than, J to I with I greater than J. Diagonal jumps where the energy is conserved, or cooling jump. And what's shown here is the rates of these types of processes. And so here, these rates are as shown as a function of frequency and time. So this means that they are non-Markovian in the language of the theorists. And so if you take the plot here, then the yellow plot here, this shows the non-Markovian behavior where this rate, gamma, goes negative. But if we come out here into the long T limit, then everything is positive. And then in that region, we can obtain our final decay rates. And so these are rates that we then use to estimate what the effect of phonons on these trajectories of absorption and of a single photon is. And so here's one trajectory where so this is only half of PS2. And there's a pulse coming in, shown here in the gray. And so that's a pulse coming in. Now, what's shown here is the dynamics up to a certain time here, at which the first jump will occur. And the jump locations, so, and then what's shown on the top left here is the state at that time. So in other words, this tells you that the initial state of excitation is somewhat delocalized, spread over the core complex and the antenna system. And then a jump happens. And then what we've indicated here in this trajectory is the position of all the jumps from then on out. And these jumps are color coded according to whether they're energy conserving, which corresponds to a dephasing type of interaction, or whether they are losing energy, cooling, or whether they're heating. And so these are the jumps are labeled here. And each of these jumps is centered on a particular chromophore. And then if you look at how the change in populations occurs, you see here that we see that there are these steps as the population changes according to each of these jumps. So this is illustrates the basic idea of a jump being a discontinuous change in a quantum state due to some measured process. And in this case, the measurement is basically whether or not there is a photon being emitted at each of these times. And what we see is a gradual change of population from the green. So, okay, let me, so the variance, okay, so there's these dephasing jumps will actually act to smear out the energy. And then once you go into an eigenstate, then you move from one eigenstate to another. So these are kind of relaxation and excitation events. But what we see is, if we look at the center, you see then we have with time increasing, we have a transfer for population from, first of all, an excitation up to the green and blue, a small amount of yellow, but then gradually the green goes down, the orange comes up, the blue is waving around. If we go out to longer times, we then see that the yellow goes down, 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 but then at the end there's a jump and the yellow ends up on the reaction center there, and there's nothing anymore in the antenna system. And the state on which the yellow one ends up is actually the chemically active chlorophyll in the in the reaction center. There's it's not completely localized there. There's a very small amount of population on a nearby site as well. So this is very nice. This is all theoretical, of course. We don't yet know. We're starting to think about how we would actually measure a phonon jump. That's an interesting question. But here is a first result from these experiments with Graham and his group, which are on a slightly simpler system, which is the LH2 system, which is a ring shown here on the bottom left. It's part of the antenna structure of purple bacteria. And these small rings are LH2 rings, highly symmetrical. And these are absorbing excitation, which then gets transported to the bigger rings, LH1, which are linked to the reaction center. So what's shown here is just one of these smaller rings. And this is a beautiful system to test this idea of photon counting in absorption and fluorescence and correlating the two of them. Because the two rings here, one of them absorbs at 800 nanometers and the other one fluoresces at longer wavelengths, 850 nanometers, and you can separate these. And so this is the result of this is actually an experiment that was started in Graham's group probably about a year ago when Changwei Li arrived. And this is, so the idea of it is what I showed before, except now it's somewhat simpler, that we only have 2 detectors rather than the third one. But Changwei has worked out ways to filter and to separate the transmitted fluorescence from the side fluorescence. So with this experiment, we wanted to make this first pass at answering whether or not quantum jumps in absorption can occur or do occur in light harvesting systems. And so this is the kind of picture we had schematically in our minds about what would be the distribution of the number of photons absorbed for each photon that was emitted by fluorescence. And this is if there are quantum jumps, whereas if there are no quantum jumps, and each photon produces just a very small amount of excitation, and then you have to produce more and more, you have to absorb more and more photons to get excitation, we would expect this kind of much broader distribution. So the preliminary answer to the question is yes, photon jumps do occur. So that's maybe a little bit glib. This is a very, very preliminary result, probably only about a week old. And there's a lot of work still to be done, because if you look at the number of photons here, it's very high. And so this data has to be still analyzed in great detail, both for experimental, to simulate all the experimental imperfections, but also we have to understand the relationship between radiated decay of the LH2 and the non-radiated decay, which will distort a very highly idealized plot like this. But this is really very exciting. It's really the first result where we can think about learning something about the way in which absorption of the energy required to release a single electron, the way in which that happens. So this is, don't know how I'm doing. So I think I'll just mention this very briefly. I could go into it more if people are interested afterwards. You might ask, so, well, this all sounds very nice, but I can lower the intensity of my classical laser light and I could do the same experiment. So what's all the fuss about using single photon sources and building these very expensive sources of pairs of photons? Why do I have to do all that? Well, if you take a coherent state, which is a state of laser light, then on the top right is shown the distribution of the number of photons that you expect from a coherent state. That's a very well-known distribution. And the orange lines are the ideal Poisson distribution, which governs the population of single photon, 2 photon, 0 photon, 2 photon, 3 photon states, and so on in laser light. And if you now look at these trajectories that we were developing, this is now just for 3 sites and only 1 mode of light. If you look at the dynamics now conditioned on not having seen a photon by time t, if you have a single photon state, you will, after a certain time, if there's no photon being seen, you will know for sure that that state system is in the excited state. So then 1 minus the probability of the ground state will be 1. So this blue line goes up. This blue line with conditional dynamics shows a clear signature if you are still in the ground state, or if you are in the excited state, in this case, after your photon pulse here has passed. Whereas if you put in a coherent state and you do the same calculation, namely a conditional dynamics where you're asking about the probability of not having seen a photon by time t with a coherent state, you see the line down here, the blue line below the orange line, which is essentially identical to the unconditional dynamics. And basically this means that you have absolutely no information about the number of photons that were producing this signal here. And if you look at the counts you actually observe, you do find then with the coherent state that you are reproducing in terms of the counts that you're observing. You're not always observing 1 count, you're observing a Poisson distribution of the counts corresponding to the average number of photons in your pulse. So this is very clear evidence that you cannot see, you cannot learn anything about the conditional dynamics if you have a coherent state. And this is similar, that basically if you're just looking at populations, there's very little difference between a single photon state and a coherent state. So you don't learn anything from doing the usual kind of spectroscopy in density matrix picture. So I'm going to quit this. I think yes, I'm about out of time. So let me just summarize. So what I've shown you and really emphasized is that we have certainly already beyond the fact that we're in the second era of quantum biology, where we are looking at phenomena that really involve non-trivial quantum effects, quantum dynamical effects, like tunneling, and also quantum correlated effects, entanglement and coherence of different kinds, and trying to understand the sources of the coherences. That for doing this and unraveling the dynamics, we really have now a new probe with quantum light sources. And in particular, for the example I've shown you for looking at light harvesting, the use of heralded single photon absorption and fluorescence gives us insight both for non-trivial aspects of the absorption, namely whether or not we have quantum jumps, but also, as I showed you, this distribution can give us a new way to look at coherences in excitonic energy transfer. So that's these 2 points here. And there's a strong indication from the new results from Graham's group that there are indeed quantum jumps in absorption. But clearly there's a lot more work to be done here. We have to do, in particular for the first thing we're going to be doing next on the experimental side is to do detailed modeling of the herald photon count, taking into account the radiative, non-radiative rates. And then we would like to include, theoretically, we're including electron hole pair generation, theoretically, that's already underway. And experimentally, so eventually, this is not within the next year or two, eventually we would like to measure not just fluorescence as a proxy of electron hole pair transfer, we would like to measure single electrons. And down the road, this will be possible because people are already measuring single electron transfer from photosynthetic systems in other situations. And this kind of work will allow us to analyze the role of vibrations and phonons in this whole transduction of the light energy to electron hole pairs in more detail. Quantum jumps is a very interesting topic. So here I could mention several things. So one is it's very interesting to think about, well, what would be a manifestation or what would be a detector of a phonon jump, the kind that I've shown you. Another point here is very fundamental, that quantum jumps are discontinuous only if you know nothing about what's happening. But it was shown last year, and beautiful work from Yale, that if you can go in and you can look at the conditional dynamics experimentally more carefully, you can actually follow a quantum jump as it happens. And any quantum jump that's completed turns out to be deterministic. So the only quantum jumps then that still remain as discontinuous and completely unknown and stochastic are the jumps that never really fully happened. So that's a bit of a mind bender, but that's, I think, a very exciting direction to think about for the future. Of course, that kind of experiment is still very hard to do, even on an isolated atom, let alone in a photosynthetic system. But now let me come to the implications for biology. So there's a lot of implications here for photosynthesis, in particular for the energy budget. The original question that I posed, and then for the microscopic analysis of quantum yield and the robustness also of photosynthesis. And looking ahead, and this is again further down the line, it's great to think about the new tools that might come over the next 5 to 10 years. In particular, we would love to see some tool that would give us a spatial distribution of the absorption events, not just temporal distributions. And I look forward to the work of Naomi Ginsberg and others that might eventually give us this on the length scale relevant to the light harvesting complexes. And finally, let me just quote Niels Bohr from his essay in 1933, or his first sort of speculations about biology and quantum physics. He made the statement that light is our principal tool of observation. And this is, 90 years later, it's maybe not still 100% true, but still quite a prescient observation. Namely, light, it still remains a really excellent way to probe biological systems. So with that, let me acknowledge the people who really did the work. And on the theory side in my group, this is Rob Cook, our postdoc, who's done all the beautiful work on quantum trajectories, condition trajectories, and introduced a lot of these ideas into our group. And this beautiful work with the Markovian phonons, this was all his work recently. Li Wen-ko, a graduate student who's working with Rob, and he did this a very nice analysis of the absorption probability, showing basically quite generically that it should be of the order of 10 to the minus 6. Herman Chan was a student in between undergraduate and graduate for a couple of years in my group, and he did a lot of the preliminary work on excitation with coherent states. And then on the experimental side, in this joint project for the photon counting, of course, my esteemed colleague, Graham Fleming, and the postdoc, Mike Chan-Wei Lee, who produced this very beautiful first set of results on the LH2 system, together with Kadron Orcutt, graduate student in Graham's group. And then also we envisaged this project in the beginning, planned it together with the group of the quantum optics group of Jonathan Matthews in Bristol in the UK, and his postdoc, his graduate student at the time, Javier Sabinas Chesterking. And they have been invaluable in injecting the tools of quantum optics into this collaboration. So let me then close with, this is sort of the motivating biological link to biology. So I think we're doing very well coming down on the tool side, probing to ever smaller timescales and smaller length scales. The key task for a quantum biologist is, however, to come back up on the other side. And this, I think, is a very, very difficult area. And this is where we really need input from biologists, and we need a lot of discussions with biology, because the question whether any of these phenomena that are being observed or studied theoretically, how they are relevant for biological function is I think, very much a big and challenging question for the present and the future. So this is the very last slide. So let me end with one quote from a physicist, Albert Einstein, and then another quote from a biologist, both of which indicate biology is really cool and really, really hard. So with that, thank you very much. And I'd be happy to answer questions.
Birgit?
Yes.
Yeah, it's Judith. Hi. Very interesting. I'm sure I have much to digest here, but I do have a question in your computation. When you introduce the phonons,
Yes.
the frequency of the phonons is really critical, and you didn't really focus on that. Perhaps you said it and I missed it. Could you just comment on what kinds of frequencies you're talking about and how they relate to the chromophore as opposed to the protein environment?
Okay. So what's this? I'm going to go back.
I mean, there is this question of whether phonons, in fact, would impede the behavior rather than facilitate the behavior. So that's
let me show you this. So this is the antenna this is actually specifically, so the calculation that Rob did has phonon, what are called phonon densities of state. So if you look at this theory expression here, right? So for each site, for each chromophore, there's an interaction with that's linear basically in the phonon coordinate as the excited state of the chromophores, essentially electron density increases, so it pushes out and it causes a phonon a displacement of a vibrational coordinate. So that's the reason why this is linear. Okay. Right. And then so because each site has a whole set of vibrations with which it interacts, and those vibrations with which it interacts could be local intramolecular vibrations in the chlorophyll, to which will be very strongly coupled, or they could be sort of skeletal vibrations of the protein scaffold, in which case there's generally weaker coupling. But this set of modes here will give rise to some density, which is known as a spectral density, which is the analog in the Fermi golden rule of this density of states here. And what's shown on this slide is the result of those spectral density, which are calculations that people make. That there's measurements from fluorescence line narrowing. You get some experimental input for that, but people also make a lot of molecular dynamics types of calculations nowadays to calculate those spectral densities to see exactly which kinds of frequencies coupled to each of the sites of the chromophores. And so this figure on the right shows such a set of spectral density calculations and then translated into, so the spectral density calculations give you this quantity C, which is a correlation function of the particular vibrational mode at the site M here. And then you Fourier transform that and it gives you this rate. So what's shown here, and that rate is a function of frequency and time. And then if you look at this, at the long time limit of this, so this is shown on the bottom here, this is the kind of, essentially the density of vibrational states that goes into a Markovian calculation. And it has this set of sharp peaks, and each of these sharp peaks represents what one would call an underdamped oscillator, basically a strongly coupled, maybe resonantly coupled vibrational motion. This might be resonantly coupled. Let me see, take one of these. This mode here might be resonantly coupled to one particular excitonic transition. And then there would be a very strong effect on transport. And then underneath this, so in the beginning, when before people had this kind of data, they would represent the spectral density as something very smooth. Okay. But this kind, so when you see a spectral density or a set of rates like this that have these sharp peaks, this means that there's a considerable amount of molecular level detail gone into making this, and that probably most likely the data that went into this actually did look at specific vibrational modes inside. These ones are probably inside the chlorophylls, inside the chromophores. Yeah, inside the chlorophylls.
Yeah.
Okay. So I didn't talk about it, but yes, there's a lot of work that people have done on it, and we took the best ones that were available at the time.
Thank you. Okay, great.
All right, I saw Jeff Reimer raise his hand first.
Yes, thank you, Julia. Birgitta, thank you for this brilliant and lovely lecture. As others have said, my mind just sort of boggled for a moment. The thing that strikes me as most counterintuitive is the notion that the light impinging on the leaf is incoherent, is coherent rather. And yeah. I'm struggling with that. Rather than take you and this august audience through that argument again, maybe I could just ask, can you imagine a light source which impinges upon the leaf, which at the center is not coherent?
Well, so supposing you take one of these single photons, and I think most people are comfortable with a little wave packet representing a single photon. Mm-hmm. That's what we did. So think about what it would mean to have a little wave packet that's incoherent. One way you can make it incoherent is to add a phase. Mm-hmm. That would be changing, just randomly switching in time. So if you have a pulse that says 20 femtoseconds in width, and every 2 femtoseconds you make it have a random blip in phase due to whatever reason, then you do that, and you'll find that when you calculate the bandwidth of that little wave packet, it becomes very much larger. So then that's inconsistent with the notion that the chlorophyll molecule, in a sense, collapses the wave packet onto something that has caused absorption over its own native bandwidth, which is the width of that spectral line. Mm-hmm. Because if you really have a lot of, if you introduce a lot of random phases, then your bandwidth will get very large. And as I showed you in that figure about your absorption probability will just get much lower. I mean, it'll go orders of magnitude down from 10 to the minus 6 to 10 to the minus 10. And we know empirically that it's 10 to the minus 6. So I think that's all consistent. I'm not saying it proves anything, but I think it's consistent with the picture of a coherent wave packet.
Thank you. I understand now. Thank you for that clear answer.
Okay.
Okay, David Limmer.
Thank you, Birgitta. That was a beautiful and very thought-provoking talk. I want to ask a question that it might be a physics question or it might be a philosophy question. So we'll see.
They merge in places.
Indeed. And it's actually on this slide, so it's useful that this is up. So
Okay.
so when you think about quantum jumps, one can kind of get your head around this largely in the context you talked about them in terms of photon measurements.
Yeah.
Yeah, as you alluded to already at the end, when you start thinking about this in the context of phonon weak measurements, it's a little more dicey. But especially when you think about this evolving from a non-Markovian equation, when you have these negative rates, what is meant by a quantum trajectory becomes a little bit hazy, I think.
I agree entirely, but that's exactly why we avoided that. We have so far avoided that by just working here. So we took.
Okay, all right. Well, but I'm curious what your thoughts are about the interpretability of non-Markovian quantum dynamics.
Okay, so let me just say a few words about the interpretability in the Markovian limit.
Yeah.
In the Markovian limit, I know that everyone's happy with quantum jumps and quantum trajectories in the Markovian limit because the idea is that if you run a single calculation, it's not that you would be able to go and see exactly that trajectory in the laboratory, but it's representative, right? And if you sum over an ensemble and your calculations are an accurate model, then you'll get agreement with the experimental average, right?
Mm-hmm.
Okay, so there nobody has any doubts, or I think most people don't have any problems with the interpretation of trajectories in a Markovian system as having some physical reality.
Right. Yeah.
Okay, so in the non-Markovian regime, that's a different matter. So you have these negative decays here, meaning a revival of coherence, and that's exactly what we mean by non-Markovianity, right? Things come back. The bath information goes into the bath, and it doesn't just immediately dissipate and never comes back. It does come back.
Mm-hmm, mm-hmm.
So in that regime, it's hard to actually really make there are many different models for trajectories in the non-Markovian regime, as you know, and most of them don't really have a good physical interpretation. In fact, this paper that I quote here, they actually do trajectories in the non-Markovian regime.
Mm-hmm.
We avoided that though, because there's always a time zero. If you do a non-Markovian calculation, there's a time zero. You're looking basically at a decay and from a particular time.
Mm-hmm.
So these kind of calculations in this context, I mean, this is Alan Spruguzik, former Berkeley graduate, and this paper was one of his first papers in studying light harvesting as well. But he, like many other theorists then and up to now, in fact, don't normally consider the light absorption step. They start off with a system, they say, suppose the system's in the excited state, maybe with probability 10 to the -3 or something after a laser excitation, but they don't worry about how it got there. So there's no notion of a real zero time.
Mm-hmm.
Whereas for us, we have our our single photon pulse, which is setting a zero of time. And it's not clear to us, at least in a couple of weeks, if we looked at this, it's not entirely clear how this kind of timescale of non-Markovianity, how that should play out in a calculation which has a real zero of time.
Yeah, that's an interesting point.
Yeah, so that's why we've so far avoided it.
I see. Very good. Thank you.
Okay, Aran Rabbani.
Hi, Birgit. Thank you for a very interesting talk. I have a simpler question, I think, but I might have got it wrong. So you mentioned in one of your slides that the single site has a faster decay than the multi-site result, or did I get this incorrectly?
Let me just, whoops, let me see. Was that back in the...
Yes.
Here. No, this is not single site. This is, sorry, the other way around. The collective...
Yeah, is it?
The collective decays faster than the single, which is a classic...
Okay.
That's a classic behavior of superradiance.
Okay, that's what I was going to ask. Is this simply a superradiant effect?
Well, okay, superradiance means different things to different people. So we would like to avoid using that word right here. But it is a collective effect.
Okay.
It is, as you say, it is in a sense simply a collective effect. But the importance here is that this is a collective decay rate, but then when you actually construct your Hamiltonian for considering excitation of the system, these same quantities, these are basically the analog of Wigner-Weisskopf rates. These, so as in the Einstein A and B coefficients and the spontaneous emission, it's always the same underlying rate. So these rates will also control the rate of absorption. And so this means that the rate of absorption will also have some enhancement. Because of, and that's so it's not too surprising if you look at this. So here's the wavelengths of the light and here's the PS2. So as far as the light's concerned, all those chlorophylls inside this PS2, they're all sort of on top of each other, and the light is certainly viewing these chlorophylls as one collective object.
Yeah, so in principle, you should be able to see, in principle, you should be able to excite a coherent.
We do. So when we do our excitation operator is the L dagger of this. And so it has contributions from all of these ends. But the interesting thing is this is not the same kind of, so we get to delocalized state. I think I showed it on the, here. This is the initial state. This is on the left up here. Yes. The intensity of these dots indicates how much of each chromophore is excited initially. Right, okay. And so you have a considerable amount of delocalization. But unlike what happens in a semi-classical treatment of the matter-light interaction, this delocalized state is not necessarily an eigenstate of the excitonic Hamiltonian.
Right, because you're having a pulse and you're making a superposition probably. But then this means that the mechanism would be that you're exciting a delocalized state. And in fact, it's not how long it takes you to travel to the central site, but it's actually how long it takes you to relax to it from that delocalized state.
Yeah, well, but it's a combination of the two. Because
because there is some,
there's a spatial, because as the calculation proceeds, the excitation is moving. So when I go to the end here, these are the jump operators. This is the end. So the excitation has actually moved.
Okay,
so it's ended up on this site here, and there's another site very faint next to it. So it's moved to a more strongly localized, but not completely localized state, which is lower in energy, and it is in a different location from the initial excitation. But this initially delocalized state is not a. It's not a. The interesting thing is it's different from what you would predict if you use semi-classical interaction.
I see. You treat the light classical.
Yes, because it contains it's defined essentially by the interaction of the system with the light, and it has this via this Green's function approach, which I think you would probably appreciate. It has the notion of light interacting with one chromophore, but then that light emitting and then interacting with another chromophore and then coming back on itself.
I see.
So it's a true sort of collective internal matter-light interaction inside the set of chromophores. And it depends on so the effects of this will depend upon the polarization of the light. And they also, if you look at the emission, the effects will also depend upon the solid angle where you're taking this. So, and this is something that's often mentioned in small print in articles about superradiance. Superradiance in emission does depend upon the direction in which you're measuring. And so if you integrate over all angles, you probably won't see any differential superradiance effects.
Okay. Thanks.
Okay, Graham Fleming.
This is partly to answer Iran's question. There is perfectly good experimental evidence that there is modest superradiance in LH1 and LH2. I don't think the situation with respect to PS2 is experimentally clear, These experiments are not easy because you need to measure an absolute quantum yield in order to see if the radiative properties have changed or not. But there is definitely collective emission to a modest extent in those ring structures.
Yeah, I mean, Graham, would you say this is 1 order of magnitude here? I think that's reasonably modest, given that you've got 300 chromophores. So if they were all aligned, you'd have hundreds, a factor of 100. And this is quite a bit less than that.
Well, actually, you don't. I mean, even in J aggregates, you don't see anything that large,
Right.
because the phonons destroy
Right.
the superposition before the photon gets emitted.
Right. Yeah, so that's actually one reason why we wanted to add the phonons to these calculations, because we were worried that we were worried that these things, these oscillations and so on, they might actually go down quite a bit. So that's one thing we're going to investigate further with these. Now we have these phonon jumps in here.
All right, I see a question from Naomi.
Yeah, thanks. And actually, please stay on this slide because I was really intrigued by Birgitta, what you were saying in response to Iran's question. And I'm just like really scratching my head thinking about space, time, and energy here. So it absolutely makes sense to me that, just due to the proximity of like all of these chromophores in PS2 relative to like wavelength of visible light, that initially you do have something like delocalized over all of them, and presumably like the energies initially are close enough together and the absorption event is so fast that like you're accessing all of them in some capacity. But then in order to get from there to being able to say that this excitation has traveled, I guess I'm just wondering, like, what's the sort of time ordering, if you can talk about time ordering of like localization and like subsequent travel? Like, how do I think about the dynamics of that process and the connection between the localizing and the sort of centroid changing, and how that also relates to this, like the sort of spectral properties, if they matter at all, of the different chromophores?
So there's a lot of things in there. Again, these results are also about 5 days old too.
We're helping you think about them.
Yeah. So no, but we have right. So here's a few things I can say. So firstly, in this case, these are the initial chromophores that are excited. These ones here, and so they're primarily in this. The wavelength here was centered at the median energy of all these 150 something sites. Mm-hmm. So it was probably still in the LHC2, but there was some overlap with the core complexes, because the energies sort of go, there's a sort of hierarchy goes down, roughly speaking, from the LHC2 to the core complexes to the reaction center. Okay, so, but I think what this tells us is, because of the way we've handled these collective excitations, these initial pictures that I showed of PS2 with the white arrow are misleading, this one. So I showed this picture here, and this is kind of like the standard picture that one says, oh, well, there's an excitation somewhere in one of the LHC2s, and then it has to travel through some complicated path. Maybe it's random, maybe it's not, until it hits the special pair in the reaction center. And I have indicated here with some white cloud how the usual picture that one has is that there's you excite to some excitonic state of the antenna. And that excitonic state, we know, it's not delocalized over the entire system, but it's localized over a small number of chromophores. And of course, if you have a broadband pulse, then you're going to have, where's my pointer. If you have a broadband pulse, you'd have several of these, so you might actually then extend over multiple regions. Okay. Our excitation operator is a little bit different. As I said, it's really it's forced to be that way because it's a single photon, and it's treated quantumly as a single photon pulse. So that's why it's not an eigenstate. It can be decomposed into sum of eigenstates, but it's not actually produced. Even if we had an infinitely narrow bandwidth pulse, it would not create one single eigenstate. Okay. So then how it moves, well, and this is something we now are going to have to analyze, is if we as a function of time, we decompose our state and we see which eigenstate it's in, then we might be able to analyze it a bit more carefully. Another thing we can do is, if I go back to the definition of the jump operators, the phonon jump operators here. So these energy jumps, which are called diagonal, these correspond to dephasing types of transitions. These are jumps which you see here, they don't change the energy eigenstate, and they're localized on a site. So these diagonal jumps tend to localize you on sites. The heating and cooling jumps move you from one energy eigenstate to another. And so the heating and cooling jumps tend to spread you out because the eigenstates are in different regions, right? So that's how you typically would get relaxation down. You go from an eigenstate which is localized initially in the antenna, and then the next one's a little bit further over with the overlap with the CP, and the next one's further over until finally you get to something that's lower in energy and also localized in the reaction center. But these diagonal jumps actually cause you to be localized in one site, and they turn out to be very important. So these are like dephasing, and Rob's run calculations where in the CP and in the reaction center, these jumps are absent. In fact, the reason he did that was because the so-called best spectral densities available for those complexes, which aren't as well documented as the LHC2, they actually don't have these, they don't have diagonal jumps. They don't give diagonal jumps, and that's because of the way the spectral density goes to zero frequency, which is, this is zero frequency. And so what we find is that if you don't have these diagonal jumps, then your excitation will get stuck. And you need you need these, I mean, this is well known. I mean, Paul Graham can say a lot about this. You need the dephasing to jiggle around your energy level so you can get stuck and you don't get trapped by unitary type or either unitary or just relaxation type of dynamics. Because relaxation here is the relaxation and heating are all between eigenstates.
Right. Yeah, I guess what I was confused about is at like an earlier point in your explanation, I thought, oh, it kind of sounds like you're saying, you know, you're sort of you're moving through this series of states and you're sort of projecting one onto the other, and that eventually like moves the centroid of the wave function closer to the reaction center. But these actually like keep you, like they collapse everything to a given point.
Yes, let's see, on that trajectory, let's see, this one here. Okay, this is a zero energy jump, and it actually causes it's basically can be identified as a coherence transfer. I don't know if this really helps. I mean, this is what we all have to, we still have to sort out a lot, and we are planning to go through one of these trajectories and see actually what is the spatial extent of the wave function as you go through, and what are the components and so on. So these are really all very very good questions.
I almost made a movie, but I ran out of time.
All right, Rob,
I'd love to see it.
Rob should make a comment here. I mean, Rob, do you have something to add to this?
Quantum mechanics is hard.
Yeah, well put. Thank you.
Okay, well, I don't see any more questions coming in at this time.
So Julia, am I able to share the screen? Is that what's next?
I can
Yeah.
share. Yep, I stopped sharing.
Okay, let's see. Julie, am I sharing or not?
No, not yet.
Okay, I seem to have lost my sharing capacity from the screen. Any way you can help me out?
Well, you're a co-host, so you should still have capacity.
It should be on the bottom.
Yeah. Yeah, it's disappeared. So that's odd.
I'll try undoing it and redoing it and see if that helps.
Because there is a slide I'd love to show you.
Do you want to try again now?
Yeah, it just seems to have disappeared. I no longer have the ability to share the screen, so I'm going to have to explain this to you.
You can send the slide to Julia or to me.
Well, it's not that fancy. But so I want to thank you once again for the fascinating, enlightening, and truly historic Lewis lecture. So before we let you go, what you're not seeing, but you'll have to visualize, is a piece of history from the College of Chemistry. So on the screen that you can't see is a paperweight that's in the shape of a Berkeley golden bear, which of course is our mascot, which was carved from the original soapstone laboratory bench in Gilman Hall, not too far from your current office, which is where Glenn Seaborg worked as a research assistant for G.N. Lewis. So a little bit of history, a little bit of Glenn Seaborg, a little bit of G.N. Lewis will soon be coming your way.
Wow.
So maybe it's better that you can't see it because that will just increase the anticipation. You don't have to come and pick it up. We'll send it to you.
Is it heavy?
No, it fits in the palm of your hand.
Okay. All right. Thank you very much.
It is a priceless and unique piece of the College of Chemistry.
I'll treasure it.
Glorious history, which you have now just contributed to. And by the way, in addition to that, we'll also be depositing a small quantum of research funding into one of your research accounts.
Love it.
Thank you again, Birgitta. It's a shame we couldn't do it in person, but this worked out very well. So a virtual round of applause. So Julia, what's next?
That's it, unless people want to stick around for other questions. I'm going to go ahead and stop recording so you can come ask all those questions that you didn't want recorded now.